Now we're going to talk about animal diversity. So animals have over 35 phyla, or at least 35 known or identified phyla, and 34 of them are invertebrates. So most of them are invertebrates, and one of them just contains vertebrates. So not even all of that phyla is actually a vertebrate. So most people, I think, when they think of animals, they think of dogs, cats, things like that, and of course they're vertebrates. But most of the animal phyla are not. There are two major groups. One is called a metazoa or parazoa. This is a group that has no true tissues, and mainly we're talking sponges here. And then there's the eumetazoa, which are all the other animals, and that they have true tissue layers that we talked about previously. So let's just look at this. This is a diagram that's in your textbook, and it is talking about the animal groups, and hopefully it's in better focus than on my screen here. If not, check out the figure 32.11. So this is talking about the metazoa, all right? So it's saying sponges, okay? And what I like about this one is it has little pictures here that are showing you what these guys look at. And then everything else is eumetazoa. All right, so we're talking about all of these groups are eumetazoa, all right? And we are going to cover most of the ones that are in this diagram. And the other thing is that when you're talking about these two groups right here, these guys have radial symmetry, and they are basically what we're talking about is cnidarians here, which are corals, and the ctenophoras, which we won't really talk about in lecture, but we will talk about them in lab. So I gave you guys a handout on that. All right, so those guys that have radial symmetry. Everything else is bilateral. So everything from here on out is bilateral, and so I'm just kind of showing you how to read this diagram here. And then we have what is known as protostomes and deuterostomes. The deuterostomes include the vertebrates, but also starfish and the let's see, Hemolatoda. I'm sorry, I don't have my glasses on here. And anyway, these guys right here, these are all deuterostomes. Remember, we went over this in the last lecture. Basically, their body formation or their pattern of development is quite a bit different than the other groups, which are protostomes. These guys are the bulk of the organisms that we are going to talk about. And then we will also talk about these couple of groups as well. So just take a look at that diagram, and we'll go talk about it more as we go along. All right. So we're going to also, I wanted to also remind you that we were talking about before that the choanoflagellates, which gave rise to fungi and the animal groups, one of the reasons why they think that is because these sponges have cells that look really pretty much like that. All right, so these are the beginning or the first type of animals. They're very simplistic, and then all the other animals were thought to have been derived after that, or coming from that common ancestor. All right, so invertebrates then make up the bulk of what we're going to be talking about. And so we're just going to go through several of the groups. By far, not all of them. I think we're covering like 9 groups or so. I gave you a chart that you can fill out, and I would recommend trying to get something for everything in that chart. So I try to fill it in here. If I miss something and you have a question about it, just let me know on the Zoom or in class. All right, so the Phyloporifera is the first one that we're going to talk about. These are the sponges. So they are considered metazoa or parazoa. That's because they have no true cell layers or tissue layers. The term porifera means pore bearer. These organisms are sessile, meaning that they are attached to a substrate of some sort of rock, or even like a piece of coral, another organism like coral or something like that. They have very porous bodies, and they don't have any symmetry at all. So they, sometimes look like they have radial symmetry, but mostly they are considered asymmetrical. They have what is known as choanocytes. Choanocytes are called collar cells. I'll show you a picture of that in just a few minutes. So that, that is very similar to the choanoflagellates that we talked about before. So these are the ones I'm talking about. But what I wanted to say here is that they are suspension feeders. What that means is they are hanging their flagella out in the water. The water, as it washes by, has food suspended in it, and it will grab that food source. All right. Now, there are three basic types of sponges. You'll see in lab that there's other groupings that we can do. But basically there's a type that contains no spicules. So what spicules are is they go throughout the body. They're not really a skeleton, but in essence they kind of act like one. They keep the sponge somewhat rigid in its environment. So there are some that have no spicules. They just have what is known as spongin material that is sort of giving it some shape, which is very squishy. It's like the sponges that you use at your kitchen sink. Then there's the silicia. These have silica oxide, so they have glass on the inside of them. We have a really nice one in the lab that you can look at. And then there's the calcarea, and these guys have calcium carbonate as their spicules. So there's those three basic types of spicules that can occur. They have three layers, even though they're not true tissue layers. They have an outer layer, an inner layer, and then some jelly stuff that's in the middle. The outer layer is called pinacocytes, and they are covering the outside. Then you have the choanocytes, which again we're going to look at in a sec, that are covering the inner part, and then you have the mesoglia. You also have amoebocytes that move around in the mesoglia mostly, and they are going to carry nutrients, and sometimes they produce the haploid cells that act as gametes. All right, so let's look at the organism. So looking at the organism a little bit closer, of course there's a name, so let's just first look at this choanocyte here. All right, so you have the choanocyte. This is the cell body of the choanocyte. It has a little collar, and that collar has a flagella that is rotating, and it basically circulates the food into the collar, where food can be absorbed by endocytosis. So these are the main feeding cells, and they are lining the inside part of the sponge. The outside part of the sponge has a lot of holes in it. And so the porocytes are the holes that are going through. The mesoglia is like the jelly stuff that is in between. You have the pinacocytes, which are the actual cells that are on the outside. In this case, they're calling it the epidermis. And you're going to have water that is flowing into the sponge and out of the osculum. So the osculum is kind of like a mouth, except for the water is flowing out instead of coming in. So it's not like we normally think of as a mouth. This is also showing you the amoebocytes. Again, they travel around the organism. And let's see, I think we talked about everything that is on this diagram. All right. Okay, so the other thing is we're going to start talking about the reproductive and the different types of reproductive, and we'll do it as we go through. These guys are hermaphrodites, which means they can be both male and female. Usually they kind of alternate that. And they have, they're producing haploid cells. And those haploid cells will either go out in the water, or perhaps even in the water within the sponge itself will create a zygote that would be moving around in the mesoglia. But eventually that's going to make a flagellated sperm, and a flagellated larva, and that flagellated larva will then swim to a new substrate and start a new sponge elsewhere. These guys can also produce antibiotic substances or defense compounds. some of which can be used by us as a perhaps source of, I think they make a cancer drug out of it. They use it to help produce some antibiotics. So, sponges have some uses other than just wiping up water around your kitchen table. Okay, so the next group is called Eumetazoa. So this is, again, the big, large group that contains all the other organisms or all the other animals that we're going to talk about. These guys all have true tissues, and they, the first group of them are also in a larger class or a larger group called Radiata, meaning they have radial symmetry. So the first phyla here, then Cnidaria, has radial symmetry. The name comes from the term cnidos, which means stinging nettle, and that is referring to cnidocytes. Cnidocytes are stinging cells, and I'll show you a picture of that in just a second. Some representatives of this group are jellyfish, corals, and sea anemones. And there are two forms in which they can exist. One is called a polyp, which is the sessile or attached to a substrate organism. And then the other form is a free-floating form called a medusa. And the medusa floats around, usually mouth down. All right? So these organisms are diploblastic, meaning they have two cell layers, and they have radial symmetry like I mentioned before. All right, so first of all, let's look at this idea, or this cnidocyte type of cell that exists on these organisms. All right, so if I was looking at a hydra, which is what this is a picture of, lining the hydra tentacles that are coming off are these little cnidocytes. So the cnidocytes are the actual cell. They have a pneumatocyst, which is kind of like the capsule that includes the trigger, and the trigger will, once it's touched, will set off this coiled thread out, and it will sting its prey. And sometimes the stinging is quite dramatic or quite toxic. So we'll talk more about some of the types of organisms, but those of you that grew up in Florida, you know about man-o-war. So man-o-war would have these cnidocytes. All right. Now here's just a representation of the polyp and medusa stage. So again, the polyp is sessile. It's going to be attached to something. And it has the main body part. It has the two, it's diploblastic, so remember, it will have an outer cell layer called the epidermis, and it will have an inner cell layer, which is called the endodermis, and then it will also, they didn't really put on this diagram, I don't see, and then they have the mesoglia, and the mesoglia is kind of like the jelly stuff that you saw in the sponges as well. All right, so this is a place where the polyp will have one mouth opening, and it acts as a mouth and an anus. In other words, the water will flow in and around and feed all the cells that are on the inside and then flow out. So in this case, these organisms are receiving all of their nutrients and so forth from the water that surrounds each of the cells. And the medusa stage is the similar way, except for the mouth anus is down, and otherwise it's doing that. I just want to mention here, I forgot to say, that in porifera, they also receive all of their nutrients and get rid of all of their waste from the water that surrounds them. So these are doing everything that is in that chart on the cellular level. In other words, excretion, feeding or digestion, getting rid of nitrogenous waste, basically all the things that are in that chart, okay? All right, so in this case, these guys are doing the same thing, except for they have the two, major tissue layers. Another thing they have is a nerve net. So like what a nerve net would be, it's kind of easiest to see in the medusa. If I were to look at this from the top, the nerve net a lot of times will go as a ring and then radiate out along all the tentacles. So basically it's kind of like a system of nerves or radial sensory structure, and it helps the medusa. It also exists in the polyp as well, but it's radiating out along the tentacles, and it can help, first of all, notice if there's a food source there, so it can use its tentacles to kind of guide. And the other thing is it can help the medusa know if it's upright or not. So that means that the mouth is facing downward and the rest of it is facing upright. All right, these guys also have something called a planula larva. So the planula larva can swim. It looks kind of like a paramecium, sorta. It's kind of like a hairy oval thing. It's bigger, of course, than a paramecium. You could actually, they're small, but you could see them. And they swim around, and they perhaps will form a medusa in another location, or they will form a polyp in another location. There are two major classes. There's the Medusozoa and there's the Anthozoa. The Anthozoa exists in the polyp stage only, and the Medusozoa has both stages. So let's look at an example, and also then we'll look more closely at the classes. Okay, so this is a hydra. You can look at these hydras in lab. They sometimes have live hydras to look at. So you know, hopefully they'll have that, and if they don't, they'll have some on the slides. Okay, so in the hydra, what's happening, the planula larva, there it is, that's what it looks like. It will settle on a substrate, and that substrate then will start to grow the polyp stage. So in this case, it'll grow into a polyp. These are what they call feeding polyps. They have the tentacles, and they will, like, you know, the cnidocysts at the ends of them, and they're pulling in food via endocytosis mainly. All right, and then eventually you'll get a reproductive polyp. That reproductive polyp will then break off and become a medusa. So this is like the reproductive one. Inside the reproductive polyp, there will be gonads that produce, through meiosis, egg and sperm. Now these guys are truly egg and sperm, not just like in the periphery where they're just gametes that are out in the water, sort of unidentified gametes. But you know, they're male and female. But anyway, these are true egg and sperm. They will come together, make that zygote, grow into a planula larva, and start all over again. So that's a basic life cycle. This hydrozoan, which is called obelia, is then a medusozoan. Yeah, I said that right, I think. Okay, now this is, I put this chart up here. This is the from the old textbook, but what I liked about it that they don't have in the new textbook is they have good examples of all of the old class names, and these are still like subclass names. So the Medusozoa, of course, is our class. Anthozoa is the other class, the second one. These guys all have the polyp and the medusa stage, but this is nice because you can see, for example, the hydrozoa. Some of the things that you know here are things like some corals, the hydra that we were just talking about, man-o-war. These guys are all in the hydrozoa. The scyphozoa are jellies and sea nettles. These are the typical jellyfish that are not man-o-war that you might see around. And they mostly have the medusa stage. They have a polyp for a very short amount of time, or not at all in some species. And they will then, you know, float around, mostly in the medusa stage. And the cubozoa, these are the ones that you probably heard on the news. These are box jellies or sea wasps. These guys are very, very toxic. They can actually even kill a person. So they weirdly have complex eyes or sensory organs to help them navigate their environment. All right, so the Anthozoa is the second class. These are sea anemones and the coral, a lot of corals and sea fans. These guys are all sessile, right? That means that they are only in the polyp stage, and they are just hanging out in that polyp stage their entire life.